So welcome everybody, welcome to any new attendees, and welcome back to anybody who might have attended the seminar series last term. We're delighted to see you here today at the Cambridge SRI Early Researchers Seminar Series. My name is Aideen and I'm one of the organisers of the seminar series. As I said, we're delighted to see so many of you here today for the first series, the first seminar of Lent term. So as many of you might already be aware, Cambridge Reproduction is an interdisciplinary initiative that brings together researchers from across the university who have an interest in any aspect of reproduction, everything from science, technology and medicine to arts and humanities. And membership of the SRI is open to all staff and postgraduate students, and it gets you access to many of our events, funding schemes and regular newsletters, which have research highlights, events, opportunities and vacancies in the realm of reproduction research. So everybody's welcome to join the SRI. You can download the membership form from our website, and I think Christina, the fantastic administrator of the SRI, will be putting some links in the chat where you can find the download forms. So as I mentioned, today's seminar series is part of the Early Researchers series hosted by the SRI, and we're very grateful to the volunteer coordinators for organising this series, to all of the speakers, especially our speakers today, who have agreed to take part. So the seminar is held online on the third Thursday of each month from 1 p.m. to 2 p.m. and details of forthcoming seminars can be found on the SRI website. We're always interested in receiving abstracts for future seminars. If anybody, specifically postgraduate students, postdocs, early career researchers would like to take part, either to present their own research or to chair one of our seminar sessions, you're more than welcome. Please follow the link on the Cambridge Reproduction website to find out more about how to do that. So without further ado, I'll introduce today's speakers. So Amy Wilkinson is a research assistant in the Rug, Gun and Reich groups in the epigenetics department at the Babraham Institute. Her current research explores the use of human pluripotent stem cells to study amnion development and function. Amy completed her BA in Natural Sciences at the University of Cambridge, carrying out her undergraduate project with Dr. Erica Watson, where she investigated the effect of abnormal folate metabolism on uterine function and placental development. And today she's going to speak to us about her research entitled Disruption of Folate Metabolism Causes Poor Alignment and Spacing of Mouse Conceptuses for Multiple Generations.
Thank you very much for that really nice introduction. And thank you for letting me present my research for you all today. So what I'm going to present today is based off my work that I did with Erica Watson as part of my undergraduate degree, and has recently been published in Frontiers in Cell and Developmental Biology. So I'm just going to share with you a few of the highlights from this paper, entitled Disruption of Folate Metabolism Causes Poor Alignment and Spacing of Mouse Conceptuses for Multiple Generations. So folate, or folic acid, is an important vitamin for cellular function. And as you can see here from this diagram, it has broad effects during pregnancy alone, let alone for other processes during life, affecting from the placenta to the fetus itself and the overall pregnancy outcome. And it's been long known the link between folate deficiency and neural tube defects. And so women who are trying to conceive or have fallen pregnant are encouraged to take folate supplementation during pregnancy. And some countries have also introduced mandatory folate fortification of flowers, such as in the US and Canada in the early 2000s. And this has just also been mandated in the UK September last year. But the mechanism of folate and how it causes these disorders is largely unknown. So what does folate do? Folate is metabolized during the folate cycle here on the left. I'll just get my laser pointer. And that links to the synthesis of thymidine. So folate is important for DNA replication and cell division. The folate cycle is also linked to the methionine cycle, which provides the methyl groups for all cellular methylation reactions, including that of DNA and histones. So folate is also important for the epigenetic regulation of gene expression. So in order to investigate the effect of folate metabolism on disorders during pregnancy, the Watson lab uses a knockdown mutation in this enzyme Mtrrgt. And this enzyme ensures the continuation of both the folate and the methionine cycles. And these mice exhibit widespread epigenetic instability. One way this manifests itself is in a range of developmental phenotypes at midgestation, so E10.5 in the mouse. We see a range of growth defects, such as growth restriction and developmental delay, as well as congenital malformations, such as heart defects and neurotube defects, and placenta defects, including this unusual phenotype where the placenta is eccentric within the implantation site. And also, as you'll probably notice from the genotypes indicated on these pictures, we also see these defects in genetically wild type animals whose maternal grandparent was heterozygous. So this indicates there's transgenerational inheritance of these phenotypes. And on top of this, it was previously shown that the growth defects, so growth restriction and developmental delay, occur dependent on the uterus, whereas the congenital malformations occur independent of the uterus and are likely arise through an embryo defect. So following on from this work, the main aim of this study was to understand how defective folate metabolism due to the Mtrrgt mutation influences early development and the uterine and embryo-specific effects involved. So first, we wanted to explore the appearance of these developmental phenotypes at an earlier stage of development to start to get an idea of when they originate. So in order to do this, we have our control group, which are the C57 Black 6 strain, as the Mtrrgt line was back crossed into this strain, and it negates the effect of the transgenerational inheritance. And then also we examined homozygous mutants so as to see the most extreme effect. So performing these crosses, we collected the conceptuses and phenotyped them at E8.5, so 2 days earlier than previous. And what we see is that a range of these developmental phenotypes are also observed at this early stage. So while most of the control conceptuses were phenotypically normal, only 42% of Mtrrgt conceptuses were phenotypically normal. 30% of the rest were developmentally delayed, 20% were severely affected, including neurotube defects, as well as the eccentric placenta phenotype, which occurred in 12% of homozygous mutant conceptuses. So this shows that these defects likely originate earlier than E8.5, but we didn't see heart defects at E8.5, suggesting that these arise between E8.5 and E10.5. We also see a low frequency of twinning throughout gestation in the Mtrrgt line, as shown by these pictures here of 2 placentas within one implantation site. And this is never seen in controls. This is thought to be dizygotic twinning, as we often see both male and female embryos occupying the same implantation site, suggesting there is a defect with how the embryos space at implantation. Now, not much is known about the exact mechanism of embryo spacing in the mouse, but it occurs in 3 broad stages. So all of the embryos enter the uterus as a cluster. They then spread in one direction by uterine muscle contraction, and then space out in both directions through embryo-uterine signaling to their final positions. But when we examined gross spacing in both groups, we see that there is no spacing defect and they are equally spaced within each uterine horn. Just to say, it's perfectly normal to have an imbalance of the number of embryos in each horn in the mouse, and we see that in the controls too. The important thing is that the spacing within a horn is equal, as we see here. So given that and the fact of the low incidence, we never see it more than once in a litter, this suggests that this is caused by an embryo-specific defect. We next wanted to examine the eccentric placenta phenotype in more detail because it's quite unusual and it occurs at a high incidence at E8.5. So just to give a bit of background of placenta development in the mouse. At E6.5, which is just after implantation, the embryo consists of cells that will give rise to the fetus and two cell populations that will give rise to the placenta. The extraembryonic ectoderm goes on to form the chorion by E8.5, so the stage that we're looking at. And at this stage, we see the allantois extending to attach to the chorion, and that will form the umbilical cord. The ectoplacental cone, here on top in light blue, goes on to form the junctional zone of the mature placenta at E10.5 around mid-gestation. And this is responsible for the hormonal production of the placenta. Whereas the chorion goes on to form the labyrinth zone, which is important for exchange between the mother and the fetus. And the placenta is in contact with the maternal uterine tissue, which is known as the decidua. It undergoes a specialized reaction upon implantation. So when we looked more closely at the implantation sites we collected, we saw that the majority of controls were perfectly aligned within their implantation site. So here we can see the ectoplacental cone in a blue through TP-BPA in situ hybridization, and the chorion here at the bottom. But we saw that a proportion of homozygous mutant conceptuses were skewed within the implantation site here on the right. We also saw them aligned as well. So to quantitate this, we measured the angle relative to the axis of the uterus. And we see that there is overall an average higher chorion angle and ectoplacental cone angle in the mutants. But you can also see there is a higher degree of variability, suggesting there's a spectrum of severity of this phenotype. And so we defined skewed placentas as those that had a chorion angle or ectoplacental cone angle greater than 2 standard deviations above the control mean, which is seen in red here on these graphs. And so when we do that, we find that 50% of GT conceptuses are skewed. Now that's much higher than the incidence of eccentric placenta classification based off gross dissection, suggesting that only the most severely skewed are identified in that analysis. We then correlated these angles, and in the GT conceptuses, we see that they have a positive significant correlation, suggesting that they're both the ectoplacental cone and the chorion are skewed to a similar amount. And this may indeed extend to the entire embryo. So a bit of background on how embryo orientation is established in the mouse. So the blastocyst at implantation is found in this orientation, so that the inner cell mass, which goes on to form the fetus, is on the opposite side to the attachment site. Notably, this is upside down compared to humans. The uterus then forms a crypt, which guides the embryo down in the same orientation. And by E6.5, the embryo is fully embedded and the uterus has undergone decidualization. The uterus axis is split into the anti-mesometrial side on the bottom and the mesometrial side at the top. And on the mesometrial side, we have the spiral arteries incoming from the main uterine blood vessel. And so in this way, with the conceptus in this orientation, you can see that the placenta will form on the side that has the best access to the maternal blood. In terms of mechanism, there's not too much known about how the embryo orientates itself, but the previous implications have been for maternal factors, such as how the crypts form, the perfusion of the implantation site with blood, and the extent of decidualization and shape of the implantation site. And so we next wanted to look at the effect of maternal genotype on conceptus skewing. So to do this, we took 2 heterozygous mice and crossed them to produce the full range of genotypes. And these gave us our mothers, which we then crossed to control males. So we're only looking at the effect through the maternal line. And then collected the embryos from these crosses at E6.5 for analysis, so that we could see the extent of decidualization and we could see the whole embryo orientation. And again, we see skewing at this earlier stage at E6.5 in all genotypes. So this suggests that the phenotype may even occur as early as implantation. When we quantitate this similar to E8.5, again, we see higher angles, though this is not significant because of that huge variability and the spectrum of severity that we see. So defining a skewed conceptus as before as 2 standard deviations above the control mean, we see that homozygous mutants derive, well, heterozygous conceptuses derived from homozygous mothers have a 57% skewing rate compared to the wild types, which are 60%. So it's all to a similar extent, suggesting that it's not a direct effect of the maternal genotype, but is actually linked to that of the maternal grandparent, which has been shown before for the other developmental phenotypes. So next we went on to look at factors in the decidua that might be playing a role in this mechanism. Through gross analysis of the decidua, its weight and cell morphology and the blood sinus area, we don't see any difference between the Mtrrgt line and the controls, but we do see disruption in some signaling pathways involved in decidualization. So just as a quick overview of the molecules involved, we see that progesterone receptor, which is really key in decidualization, is downregulated, but only in heterozygous or homozygous mutants, suggesting it's a direct effect of the allele in the maternal uterus. This is mirrored by NR2F2, which is downstream of progesterone receptor. But when we look downstream of that at BMP2, that is also downregulated in the wild type mutants, suggesting that this occurs through a transgenerational mechanism also. While it's not possible to relate this to skewing in this case because of how the analysis was done, it indicates work for future studies to assess this relationship and what else the implications of BMP2 regulation might be. Now, this second parallel pathway does not seem to be disrupted in the mutants. So we next want, given the implication of transgenerational inheritance of this phenotype, we wanted to assess this more rigorously. So we reanalyzed previously published data from the E10.5 stage, looking at different transgenerational pedigrees. So we start off with a heterozygous maternal grandmother and cross that to a control male. This produces wild type and heterozygous offspring, which we then cross to controls again to get our second generation of wild type and heterozygous conceptuses. We then carry on with the wild type generations as well to get the third and the fourth wild type generations and assess all of these conceptuses at E10.5. We then do a very similar scheme, but the only change being that the maternal grandfather is the heterozygote rather than the grandmother. And just to say, a caveat of this analysis is that we are just looking at extreme skewing of an eccentric placenta rather than the whole range of skewing in this analysis. So as mentioned before, this was never seen in controls. Whereas in the maternal grandmother pedigree in the second generation, we see 5.1% incidence of eccentric placenta formation. And in fact, this one is even completely upside down within the implantation site. If we look at the wild type heterozygous generation, second generation, it's the same percentage, suggesting that, again, it's not a direct effect of the maternal genotype, but related to the grandparental generation. Similarly, with the maternal grandfather pedigree, we see a significant 2% incidence in the wild type generation and 2.3% in the wild type heterozygous generation. So this is slightly lower than the maternal grandmother pedigree, which might suggest different mechanisms in how they establish themselves. Now, this doesn't occur through the paternal line, only through the maternal line, because when we look at a heterozygous father with a control female, these offspring only have a 0.7% incidence of eccentric placenta formation. However, as you can see, this male also corresponds to the male in the maternal grandfather pedigree, the initial grandfather. So this male does have the possibility of affecting the his daughter's uterus, which then goes on to affect his conceptus, her conceptuses. Then looking further transgenerationally to further generations, we see in the maternal grandmother pedigree that the third and fourth wild type generations maintain a similar incidence, if not increasing, suggests really clearly showing that this is a transgenerational inherited phenotype. However, in the maternal grandfather pedigree, we see declining incidences with further wild type generations, suggesting this phenotype eventually resolves, which has not been reported for this model before. So, and this also highlights again that there's likely different mechanisms causing this defect when the initial insult is the maternal grandmother versus the maternal grandfather. Next, we wanted to more directly assess the effect of the uterus, as that had not been ruled out yet. So to do this, we reanalyzed blastocyst transfer data. So using the same pedigrees as before, we take the F2 generation before implantation and transfer them into a normal uterine environment. So in this way, if a phenotype is rescued so that it doesn't appear, that suggests that the uterine environment is responsible. So this was not seen in the controls that were transferred. However, when we look at the maternal grandmother pedigree, the wild type conceptuses that were transferred still showed a 2.8% incidence of eccentric placenta formation. And similarly for the maternal grandfather pedigree, 3.3%. While these weren't significant, that was because of the low number of conceptuses that were transferred and the rarity of this phenotype. However, the fact that they do appear suggests that the mechanism is likely independent of the uterine environment. But what is the actual consequence of having a skewed placenta? We see that if just looking at incidences alone, at E8.5, there are 12% of conceptuses with eccentric placentas, whereas at E10.5, that falls to 5.8%, suggesting there may be a degree of embryonic lethality there. But looking more closely at the actual developmental phenotypes that are shared between conceptuses with an eccentric placenta, we reanalyzed the E8.10.5 data for associations between different phenotypes. So we see here matched phenotypically normal control and homozygous mutant embryos and placentas. For fetal growth restriction, there is some overlap between having an eccentric placenta and being growth restricted. However, this isn't significant. But there is a significant overlap between developmental delay and having a skewed orientation. And this could be related to the fact that the placenta doesn't form with the maximum or the optimal access to maternal blood, which may affect fetal development. We also see a significant overlap between having a skewed placenta and having a heart defect, which is quite intriguing given the recent hypotheses that there is a placenta-heart axis, so that defects in the placenta can affect how the heart also develops. Although future studies will be required to determine the extent of that in this system. And finally, neural tube defects did not overlap at all with the skewed orientation phenotype, suggesting it occurs through a distinct mechanism. So just to summarize, we have shown that abnormal folate metabolism due to the Mtrrgt mutation in mice influences conceptus orientation and spacing. And this may have implications for subsequent embryo development, though future studies will be needed to show causality there rather than just correlation. Importantly, the Mtrrgt genotype of either maternal grandparent is the initial factor in conceptus misalignment, so it is a transgenerationally inherited phenotype. The mechanism behind this, again, will require future studies to elucidate, though given folate's role, it's likely it is an epigenetic mechanism. We proposed that conceptus misalignment and twinning are caused by an embryo-specific defect, which has not been, to our knowledge, previously explored in the literature, which mainly focuses on the maternal effect. So future studies looking at this system will maybe elucidate mechanisms that are specific to the embryo in correctly orientating the embryo. And there's also the potential for these two phenotypes, so skewed orientation and twinning, to be linked. And they may both happen at implantation through similar mechanisms. And this can be backed up by other mouse models of misorientation also showing embryo spacing defects. And finally, our work highlights the importance of folate supplementation at the peri-implantation period. It could also indicate with the transgenerational aspect that folate supplementation will affect future generations as well. So unexplained pregnancy losses may actually be due to the folate status of your grandparents. And similarly, for these programs that fortify flour with folate, we may not see the full impact of that change for generations to come. So I'd like to thank particularly Dr. Erica Watson for her support throughout the project and writing the paper, the rest of the authors, and Dr. Claire Senner and Dr. Simon Tunster for their support throughout my project as well. And thank you for listening.